Hello students, myself Mrs. Vaishali Ture. I'm here to deliver a lecture for FY second semester. The title of the paper is Bacterial Cell Cytology and Biochemistry. The topic for today's lecture is Structure and Function of Bacterial Cell Inclusions, in which we shall be studying about three different granules that are produced by the bacteria, that are the metachromatic granules, the sulfur granules and the PHB granules. So let us start. So what are cell inclusions? Basically, bacterial cell inclusions can be defined as small structures or discrete structures that are observed within the cytoplasm of the bacterial cell or any prokaryotic cell. These are mainly confined to the cytoplasm, but sometimes they may also be found in the periplasm of the bacterial cell, where the periplasm extends towards the outer surface. Now, why it is necessary for the bacteria to produce these granules is because most of the times the bacteria live in environments that do not contain all the nutrients in adequate quantities at all the times. And therefore, bacterial cells, although they are simple, they have devised different methods for storage of nutrients which are employed when there is plenty of nutrient in the environment, and they store the nutrient, and whenever there is a deficiency or whenever there is a want of the particular nutrient, these granules can be utilized by the bacteria. So it is a mechanism that is employed by the bacteria in times when there is plenty of nutrient to store the nutrient and use it whenever there is a want of the nutrients. So various different types of granules are produced by the bacterial cells, and different type of bacterial cells produce different types of granules. Every bacterial cell that produces a granule or a storage nutrient storage granule, the granules are of one type. Very few bacteria have an ability to produce multiple type of storage granules. So the first type of granules that we have to study is the metachromatic granules, and these metachromatic granules are polyphosphate granules, in which multiple phosphates, inorganic phosphates, are linked to one another to form the polyphosphate. And this metachromatic granule, because it is made up of polyphosphates, it is an energy-rich molecule that is synthesized by the cell. Many microorganisms, including the prokaryotic organisms like bacteria and eukaryotic organisms like fungi, have an ability to produce the polyphosphate granules in the cells. These granules accumulate in the cell and can be easily stained by the basic stains like methylene blue because of the highly acidic nature of these granules. Being highly acidic in nature, they are stained very easily by any of the basic dyes. Methylene blue, toluene blue are the examples of the dyes that have more affinity towards the polyphosphate granules, and therefore these are the stains that are commonly used to stain the polyphosphate granules present in the bacterial cells. The polyphosphate granules, also referred to as volutin granules or the metachromatic granules, or also referred to as the Babes-Ernst granules, are the granules that were first identified in bacteria, and they were named as metachromatic by Babes in 1895. The reason why they were called as metachromatic is that these granules, they have a property of changing the color of a basic dye that is usually blue in color. As I have already said, methylene blue and toluene blue are the dyes that are used to stain these granules. So the basic dye methylene blue or toluene blue occurs as red colored when it stains the metachromatic granules. So metachromatic granules have an ability to change the color of the dye from blue to red. And because of this reason, Babes called these granules as the metachromatic granules. They're also referred to as Babes-Ernst granules because Babes and Ernst were the two scientists who first discovered these granules. And they are also referred to as volutin granules because these granules are also found to be present in the organism, that is Spirillum volutans. And these granules in Spirillum were first identified by Mayer in 1904 when he named the granules as the volutin granules. So whether it is the Babe-Ernst granules or the volutin granules, all these are the metachromatic granules. Metachromatic because they have an ability to change the color of the dye which is used to stain these granules. Why is it necessary for the bacteria to synthesize these granules? Right. As I said already, the granules are produced by the bacterial cells when there is a deficiency of certain nutrients. So these granules, the metachromatic granules, are synthesized for the requirement of energy and for the storage of phosphate when there is ample amount of phosphate available in the nutrient medium, but there is a lack of other nutrients in the medium. These granules can be degraded to form nucleotide triphosphates or to yield inorganic phosphate depending upon the need of the bacteria or the cell at that particular time. The energy that is released during the breakup of these molecules can be used for the synthesis of vital products or vital molecules like, for example, amino acids, nucleobases, sugars, as well as lipids. Because for any synthesis process that occurs in the cell, it requires energy, and this energy is utilized in the form of ATP. And ATP can be produced from the polyphosphate granules when the polyphosphate granules are broken down or degraded. Various different properties are exhibited by the metachromatic granule. The first property is metachromacy. That is, they exhibit a metachromatic effect. That is, a polychromatic stain like methylene blue, when it stains the metachromatic granule, it appears to be red in color. Under the electron microscope, these granules appear as electron dense bodies scattered in the cytoplasm of the cell. They are nothing but linear polymers of orthophosphate or polymetaphosphate of varying chain lengths. As the size of the length increases, the brightness of the color that is developed, the red color that is developed, it increases. So more the phosphate present in the granule, more darker will be the red color that will be seen. Less the amount of phosphate in the granule, the color intensity of the red color will be less when they are stained by methylene blue. Under the light microscope, these granules occur as highly refractive bodies of variable sizes. That is, all the granules that are produced are not of the same size depending upon the amount of phosphate that is present in the granule. The size of the granule is variable, right? But they are scattered throughout the cytoplasm of the bacterial cell. The synthesis of the granules starts when there is a deficiency of any particular nutrient in the medium other than phosphate. Because the granules are made up of polyphosphate, it is necessary that phosphate should be present in abundance in the medium. But if there is a lack of any nutrient, including carbon, nitrogen, or sulfur in the medium, then it leads to the synthesis of the metachromatic granules. If there is a starvation of sulfate in the medium, then the synthesis that occurs, the synthesis of metachromatic granules that occurs very rapidly, and large sized granules are formed within the cytoplasm of the bacterial cell. So, the most effective nutrient deficiency is the sulfate deficiency, which is responsible for the accumulation of massive amounts of polyphosphates in the cytoplasm of the bacterial cell. Now, when the bacteria has produced the granules, if you supply sulfate in the medium, the bacteria very rapidly starts utilizing the polyphosphate, and therefore there is disappearance of the polyphosphate granules from the bacterial cell in presence of sulfate or when sulfate is provided in the medium. Experiments have been done to see what happens to the phosphate that is present in the uh, metachromatic granules. Right? And therefore, the phosphates that were introduced in the structure of the metachromatic granules were labeled with isotopes, radioisotopes, that is 32P was used in the experiments, and this 32P was incorporated into the metachromatic granules. On supply of sulfate for the bacterial growth in the medium, these metachromatic granules started disappearing from the cytoplasm. And when studies were done to find out where the labeled phosphate was present, it was found to be incorporated in the structure of the nucleic acid of the bacterial cell. And therefore, if there is any impedance to the synthesis of the proteins or for the synthesis of the nucleic acids, the bacterial cell starts producing these metachromatic granules, which can be incorporated into the nucleic acid whenever there is the supply of sulfate in the medium or any nutrient which was deficient in the medium. The primary function of the metachromatic granule is to act as a reserve of phosphates for the cell, which can be utilized when all the nutrients are available in abundance. The formation of these metachromatic granules occurs when there is a disturbance in the production or in the formation of the nucleic acids as well as the proteins. And the granules are synthesized by addition of a phosphate from a molecule of ATP, that is the donor of the phosphate, to a pyrophosphate. Right? So, for every phosphate that has been attached to the structure or in the structure of the metachromatic granule, one molecule of ATP is utilized. Right? And therefore, ATP acts as the donor of the phosphate. Now, the ATP energy is the energy that is used for synthesis or biosynthesis within the cell. But whenever there is deficiency of nutrients in the environment or in the surroundings, the macromolecules cannot be synthesized because there is a lack of either carbon, there is a lack of either nitrogen, or there is a lack of sulfate. And because there is no synthesis of macromolecules that occurs, the ATP that is generated in the cell remains as it is, and therefore this ATP donates its phosphate to the pyrophosphate, leading to the formation of a polymer of phosphate, which is referred to as a metachromatic granule. The reaction here shows that a pyrophosphate molecule reacts with ATP molecule to form a triphosphate plus ADP molecule. The second reaction shows that if there are n number of phosphates that are attached and these n number of phosphates react with a molecule of ATP, it leads to the formation of a molecule which is larger in size by one phosphate, that is Pn plus 1, plus a molecule of ADP is formed. Right? So, the formation of the granules occurs from ATP, which acts as the donor of the phosphate. Whenever there is supply of nutrients, these granules are easily degraded, and if the degradation of the polyphosphate molecule occurs exactly by the reversal method for the synthesis of the metachromatic granule or the polyphosphate granule, then it may also provide a source of ATP energy for the bacterial cell. As we've seen for the synthesis, the donor of the phosphate is ATP. And therefore, if in the degradation process occurs by the reversal, exact reversal of the formation of the polyphosphate, then this polyphosphate, when it is degraded, a molecule of phosphate will bind to ADP, leading to the formation of ATP, and thus it can provide a source of energy for the growth of the bacteria or for the biosynthesis of the macromolecules in the bacterial cell. These metachromatic granules can be easily stained and observed by Albert's staining method. So Albert was the scientist who devised a method to stain the metachromatic granules, and the staining method uses a mixture of two stains, that is toluidine blue and malachite green. Both of these stains are basic stains, and therefore they have a very high affinity for the acidic components of the bacterial cell. Toluidine blue has a high affinity towards the metachromatic granules or the polyphosphate granules because they are highly acidic in nature, while the malachite green has a high affinity towards the cytoplasm, and therefore malachite green stains the cytoplasm and toluidine blue stains the metachromatic granules. So if you use the combination of toluidine blue and malachite green to stain the bacterial cell, right, the metachromatic granules will be observed as red colored, while the cell cytoplasm will be observed as green in color. But a mordant can be used in the staining technique, and the mordant that is used is the iodine, Albert's iodine. So basically, Albert's staining is a differential staining which helps us to stain the polyphosphate granules or the metachromatic granules with a different color than the color of the cytochrome, the cytoplasm. Right? But when you add the iodine as the mordant to the stain smear, then the color of the metachromatic granule does not appear to be red. Instead of red, it appears to be dark blackish blue color in presence of iodine. So the metachromatic effect is lost in presence of iodine, and therefore the use of toluidine blue and malachite green is sufficient to observe the red colored granules within the green colored cytoplasm of the bacterial cell. A number of bacteria have an ability to produce these granules, and these granules were first identified in Corynebacterium diphtheriae. The pathogenic organism Corynebacterium diphtheriae has an ability to produce the molecules, the granules, that is the metachromatic granules, while the non-pathogenic Corynebacteria do not produce these granules. So it can be used for the identification of the organism, whether the Corynebacteria organism is pathogenic or it is non-pathogenic. Along with Corynebacterium diphtheriae, Yersinia pestis, Mycobacterium species and species of Agrobacterium, that is Agrobacterium tumefaciens, and Rhodospirillum rubrum also have an ability to produce the metachromatic granules. Its production is used as one of the identifying criteria when attempting to isolate the pathogenic Corynebacterium diphtheriae on the Loeffler's medium. Because on the Loeffler's medium, there is deficiency of sulfate, and therefore a very rapid accumulation of the metachromatic granules occurs within the Corynebacterium diphtheriae organism. Thus, it is one of the tools that can be used for the identification of pathogenic Corynebacterium diphtheriae. The next inclusions are the sulfur inclusions. The sulfur granules are the granules that can be produced by organisms that have an ability to grow in a hydrogen sulfide rich environment. Two different groups of bacteria, that is the photosynthetic purple sulfur bacteria and the filamentous non-photosynthetic bacteria, have an ability to produce these sulfur inclusions. The photosynthetic purple sulfur bacteria are the bacteria that in which H2S acts as a terminal electron acceptor or acts as a source of electrons in the bacteria. While the filamentous non-photosynthetic bacteria, like for example Bagiota and Thiothrix, these bacteria they use H2S as an oxidizable energy source. So in one group of bacteria, the H2S acts as an electron donor, while in the other group of bacteria, acts as a source of energy. And both these groups of bacteria have an ability to produce the sulfur granules. Bacteria oxidize H2S to elemental sulfate, which accumulates inside the cell in the form of visible granules of sulfur. So, the sulfur inclusions of the sulfur granules are made up of elemental sulfur, which is usually formed from H2S because bacteria have an ability to grow in an H2S rich medium. These sulfur granules remain until the H2S source is reduced. That is, if there is a deficiency of H2S in the surroundings, then the bacterial cell starts utilizing the sulfur granules as the source of either electrons, or it uses the elemental sulfur which gets converted into H2S and can act as a source of oxidizable energy. The sulfate granules in both the groups they accumulate the granules for a transition period or a transitory period. It takes place only when the medium contains sulfide, but whenever there is a deficiency of sulfide in the medium, the H2S is completely utilized from the medium. At that time, the bacterial cell it starts oxidizing this elemental sulfur to sulfate and uses the sulfate for energy or for electron donors. If there is a deficiency of the H2S in the medium, the elemental sulfur gets converted into sulfates and the globules they slowly start disappearing from the cell cytoplasm. These granules do not usually occur in the cytoplasm. Even if they are present in the cytoplasm, very rarely or very few granules may be found to be present in the cytoplasm, but these granules mainly occur in the periplasm. As I said, there are certain granules which occur in the periplasm, but most of the granules occur in the cytoplasm. So the granules that appear in the periplasm are the sulfate granules or the sulfur granules that are produced by the bacterial cell. And when these granules are produced in the periplasm expands outwards to accumulate the granules. And when the granules are utilized, again the periplasm it contracts, right? And thus the size of the periplasm is maintained constant even if there are granules or there are no granules. The next type of granules that can be produced by the microorganisms or bacteria are the poly-beta hydroxybutyrate granules, which are also referred to as the PHB granules. These PHB granules, as the name tells you, these are poly-beta hydroxybutyric acid granules. These granules are only found to be present in the prokaryotic organisms. Eukaryotic organisms do not produce the PHB granules. Right? But why do the eukaryotic organisms do not produce the PHB granules? The reason is the eukaryotic organisms can store neutral fats in the body. But prokaryotic cells cannot store the neutral fats, and therefore they store the fats in the form of poly-beta hydroxybutyric acid granules. Bacteria that have an ability to produce these granules include Pseudomonas species, Azotobacter species, certain Spirillum and Bacillus species can synthesize the PHB granules within the cytoplasm of the bacterial cell. Many a times, only one type of reserve material can be stored in a given bacterial species. So most of the bacteria can produce either the metachromatic granules, or they can produce the PHB granules. Both these granules are non-nitrogenous granules. So, either the PHB or the metachromatic granules can be produced by the cell. Very few cells, like for example the purple sulfur bacteria, are the bacteria that have an ability to synthesize two different types of granules, which include the sulfate granules as well as the PHB granules. Right? But most of the cells have an ability to synthesize only one type of a granule at a time. Some bacteria, like for example, the fluorescent species of the genus Pseudomonas, do not synthesize any specific non-nitrogenous organic reserve material. The non-fluorescent Pseudomonas can produce the non-nitrogenous materials, organic materials, as the reserve materials in the cell, but the fluorescent species cannot develop or cannot produce or synthesize the non-nitrogenous organic reserves. So the fluorescent species can only produce the nitrogenous reserves in the cell, but non-nitrogenous reserves cannot be produced in the cell. These PHB granules can be easily observed under the light microscope as highly refractile granules like the metachromatic granules, and they are also of variable sizes and scattered throughout the cytoplasm. So under a light microscope, without staining the bacterial cell, you cannot differentiate between the metachromatic granules and the PHB granules because both these granules are highly refractile granules. The size is also variable, along with which they are scattered throughout the cytoplasm of the bacterial cell. And therefore, you need to stain the granules in order to observe what type of granules they are. They are specifically stained by Sudan black B, a stain that has an ability to stain the neutral fats. And therefore, most of the times, the poly-beta hydroxybutyric acid granules are mistaken to be the fat deposits, neutral fat deposits in the cell. But these are not the neutral fat deposits, they are beta hydroxybutyric acid granules produced by the cell. These granules are identified sometimes because of the stain, that is the Sudan black B, that imparts a color to the PHB granules as the fat reserves. The low levels of the reserve materials in actively growing cells, because the cell is actively growing, so it does not have time to just store the excess of nutrients in the form of the storage material in the cell. It is actively growing, so it is consuming all the nutrients that are available in the surroundings, and therefore, when the bacteria is growing actively, the reserve material that is found to be present in the bacterial cell cytoplasm is very less. But when there is a deficiency of a nutrient in the surroundings, like a deficiency of carbon or nitrogen, the bacteria cannot grow actively in the medium. Because when all nutrients are present in abundance, all the macromolecules can be synthesized abundantly in the cell, and therefore there is active growth of the cell. But when there is a limitation of any particular nutrient in the surroundings or in the medium, the growth of the bacterial cell, it slows down. And when the growth slows down, then the bacterial cell starts accumulating whichever nutrient is present in abundance in the surroundings. So if there is a limited supply of nitrogen in the medium, the bacterial cell will start accumulating carbon. But if there is a limited source of carbon in the medium, the bacterial cell will start accumulating either nitrogen or phosphate that is available in the medium. For the synthesis of the PHB granules, it is necessary that there should be a limited supply of nitrogen in the surrounding medium. When there is limited nitrogen but carbon and energy sources are available in abundance in the medium, at that time the bacterial cell starts synthesizing the uh, PHB granules. Right? So when there is a deficiency of nitrogen, nitrogen is required for the synthesis of proteins, nitrogen is required for the synthesis of nucleic acids. Because for the synthesis of nucleic acids, the precursor molecule is the nitrogenous bases, which include the purine, pyrimidine, the purines and the pyrimidines, that is adenine, guanine, thymine, and cytosine. All these are the nitrogen bases which require nitrogen for the formation. Right? And if there is a deficiency of nitrogen, the synthesis of the nucleic acid will stop. Similarly, proteins are synthesized from amino acids. Every amino acid has at least one amino group in its structure which contains nitrogen. So if there is a deficiency of nitrogen, amino acid synthesis cannot take place. If there is no amino acid synthesis, protein synthesis will also stop. So if there is a limited source of nitrogen in the surroundings or in the medium, but carbon and energy sources are available for the growth of the bacteria will start accumulating the PHB granules in the cell. And the amount of granules that are synthesized will be approximately up to 50% the dry weight of the bacterial cell. Till this extent, the bacteria can form the PHB granules. Not more than 50% the dry weight of the bacterial cell, but up to 50% the dry weight of the bacterial cell, it can produce the PHB granules. If there is a limitation of a carbon source in the surroundings and the medium is given is supplied with a nitrogen source like ammonium chloride, the reserve material can be utilized for the synthesis of nucleic acids as well as the proteins. Because carbon is available in the form of the PHB granules, they were synthesized because of the deficiency of nitrogen, and if the medium is now deficient with carbon, but there is enough source of nitrogen in the medium in the form of ammonium chloride, then the bacterial cell will start growing actively as a normal, actively growing bacterial cell, because the synthesis of nucleic acids and proteins will take place, and therefore the multiplication of bacteria can also take place. The synthesis of these granules, the PHB granules, occurs by the bypass of the TCA cycle, by bypassing the tricarboxylic acid cycle or the citric acid cycle, in which acetyl coenzyme A enters into the TCA cycle, leading to the formation of oxaloacetic acid. But because of the deficiency of nitrogen in the surroundings, this acetyl coenzyme A that is formed from pyruvic acid reacts with another molecule of acetyl coenzyme A to form acetoacetyl coenzyme A. This acetoacetyl coenzyme A is further reduced to form beta hydroxybutyryl coenzyme A. Beta hydroxybutyryl coenzyme A loses the coenzyme A in the SH form and thus gets converted to the beta hydroxybutyric acid. Molecules of beta hydroxybutyric acid combine with each other to form the poly-beta hydroxybutyric acid, which leads to the formation of a granule. So synthesis of the granule occurs by a process which bypasses the citric acid cycle from acetyl coenzyme A. But when there is replenishment of nitrogen source in the medium, at that time the poly-beta hydroxybutyric acid granule is degraded to form the beta hydroxybutyric acid. Beta hydroxybutyric acid gets converted into acetoacetic acid, which reacts with a coenzyme A molecule to form the acetoacetyl coenzyme A, which can be converted to two molecules of acetyl coenzyme A. Once the acetyl coenzyme A is formed, this acetyl coenzyme A can enter into the TCA cycle and lead to the uh, cyclic cycling of the carbon material or the carbon backbone for the synthesis of various different amino acids as well as the intermediates of purines and pyrimidines. Right? But the degradation of the molecule is not very easy, and it does not exactly occur by the reversal of the granule. It is necessary that the granule should be first activated for the degradation. So before we go to the degradation of the granule, let us see about the structure of the PHB granule. PHB synthesis represents a method of storing or neutralizing the acidic metabolite that is produced in the degradation of glucose or in the catabolism of a sugar, mainly the glucose molecule, which produces the pyruvic acid. Pyruvic acid gets converted to acetyl coenzyme A. Acetyl coenzyme A forms the butyric acid. Storing large amounts of butyric acid can increase the acidic content of the cell, which can be toxic to the cell, and therefore the cell can die. And therefore, the cell cannot accommodate large volumes of butyric acid. And therefore, it is necessary that the acidic charges on the butyric acid, which are usually because of the COOH group, the carboxylic group in the structure of the butyric acid. Now, this carboxylic group of the butyric acid is utilized in the formation of the poly beta hydroxybutyric acid granule, and therefore this charge is neutralized on the granule or on the beta hydroxybutyric acid, and thus because of the neutralization of the charge on the granule, the granular content usually has a less acidity as compared to the individual molecules of butyric acid. The accumulation and reutilization of this carbon reserve is metabolically controlled by enzymatic reactions, and the reactions are highly regulated reactions that occur. So it is formed from the metabolite that is on the route of the synthesis of the fatty acid, and there is a complex system which is used for the degradation of this polymer. First of all, for the degradation of the polymer, it is necessary that the polymer should be activated, and for the activation of the polymer, an enzyme requiring calcium ions is required. This enzyme, which requires the calcium ions, leads to the activation of the granule, and after the granule is activated, the depolymerase enzyme acts on the polymer and causes the breakdown of the polymer. The reaction is a hydrolytic reaction. So the enzyme is a hydrolase enzyme that acts on the polymer and cleaves the polymer to form the dimeric esters. This dimeric ester is further acted upon by the enzyme that is a dimerase enzyme, which has an ability to cleave the ester bond between the dimer and thus release the free beta hydroxybutyric acid. And this free beta hydroxybutyric acid can be further used for the synthesis of the fatty acids within the cell. The depolymerase enzyme cannot act on a polymer that is chemically synthesized and a purified polymer. That means the depolymerase enzyme is highly specific for the native granule that is synthesized within the cell. Any alteration that occurs in the granule makes the enzyme inactive on the granule, and therefore the Reutilization of the granule within the cell is highly regulated by the depolymerase enzyme, which will act on the native granule, which is not treated, not altered, but as and when it is synthesized, it remains as it is within the cell. And once the synthesis is done, whenever there is a requirement, the depolymerase enzyme can act on the granule and cause the degradation of the granule. And when the granule is degraded, free beta hydroxybutyric acid is produced, which can be used in the synthesis of fatty acids. Thank you.